Okay, thank you. Sorry for all the confusion. So we have two talks here today. One is on siRNA and gene delivery, and the other one will be on gene editing. So basically, what we're using here for this technology for delivery of nucleotides in general is a series of peptides. And the peptides that we use are amphipathic peptides. They're specifically designed to interact with different moieties depending on what kind of cargo you want to deliver, whether it's nucleic acid, cell surface, interact with the cell surface membrane. It has hydrophobic patches, so it can deliver proteins, can deliver small peptides, it can deliver nucleic acids depending on the composition of the peptide. And this is a cartoon to show what happens and how it gets through. On the right, you can see examples of complexation with the protein or peptide to form nanoparticles. On the left is examples of complexation of the peptide with nucleic acid or DNA or siRNA, etc. And the way it gets through the membrane is by not by endocytosis, but by translocation through the membrane. So this is an important advantage of this method. The type of cargoes we can deliver are varied. The number of different types of nucleotides. It can be siRNA, it can be mRNA, microRNA, plasmid DNA, proteins, depending on composition and structure. And the particle size that we create is roughly about 100 nanometers. And the peptide efficiently protects the cargo from degradation in vivo. Okay. This slide essentially shows that you could get the complex, the peptide complex, through the membrane without endocytosis, and this is translocation. So up here in this portion, what this shows is that we're looking at calcium flux through the cell wall, through the cell membrane, and when the peptides are loaded, you can see a transient flux of calcium go through, which can be measured, and then that comes back down to normal, indicating that the membrane has reorganized. And then you can challenge it again many minutes later, and you get another transient flux go through. So this way, the cargo is delivered through the membrane. There's an interaction of the peptide with the membrane components, but it essentially releases the cargo into the cell without endosomal entrapment. And you can see that here as well. This is a GAP-DH knockdown that we're doing, but in the presence of several inhibitors of endocytosis, and none of those inhibitors have any effect on the GAP-DH knockdown. So we've tested this in cell lines, of course, to begin with, challenging cell lines like embryonic stem cells and embryonic fibroblasts, etc. And the comparison here is to lipofectamine, which I guess is the positive control. But in the red is what we call the Aadigen or Aadigen peptides complex with the siRNA, and you can see a nice dose response here of the knockdown of the GAP-DH in both cell lines, and it's substantially better than what you see with lipofectamine. Also, the toxicity is much lower and very manageable in these cell types. What happens in vivo? So again, depending on the peptide, you will get a slightly different distribution. But this slide gives you an indication of it's sort of a broad distribution, and depending on the peptide selected, you see here knockdown in the different tissues. We use two different siRNA, GAP-DH and HPR1, to look at knockdown across the different tissues after a single intravenous injection. And you can see across, of course, you have in the liver, but you can get knockdown in the brain to about 40%, kidney, muscle, lung. So there's a broad distribution, and it's not all going to the liver, which is an important consideration, depending on the application. So here's a tumor model. It's HD29 colon tumor, and the target selected here is cyclin B1. And we can, again, complex with the peptide, give the tail vein injections, and then watch the tumor respond. And when you use sort of nonsense siRNA, then you don't get much of a response, or you see a dose response in the case where you're increasing the dose. What's important is that when we look at the tumors, you can see the corresponding knockdown in the tumors up to day 44. And as far as the animals, they're pretty happy. There are no significant weight loss. So this works quite well in vivo, and it manages to get to the tumor. Here what we've done is shown the concept that you could silence multiple targets at the same time, whether it's 2 or 3 targets. Here it's 2 targets. We've used cyclin B1 and CDC20, and you can see the individual effects here, and then the combination you get a additive or synergistic effect. So this concept works well in vivo for siRNA knockdown. Now, in another model, we tested a factor 7 knockdown. Some of you may be familiar with this. Factor 7 is produced in the liver. And so here we looked at sub-Q and IV, 2 different forms of delivery, but a single administration of either sub-Q or IV. And here's, we're measuring factor 7 in the blood of these animals, and we do the delivery here, and you see quite a substantial knockdown in the target of factor 7, which we can measure in the blood, both by IV, and sub-Q is a little delayed and a little bit less at the same dose, but nevertheless you can get down pretty far, almost 80% knockdown with IV, and then this slowly creeps back up. By day 70 or so, you're back to normal, and you can hit it again and once again get a good knockdown. So you can do a repeat administration depending on, again, the target you're going after, and adjust the dose, etc. Okay, so that was siRNA delivery. We can also do a delivery of plasmids or gene delivery. So this is, again, conceptually showing you that we can do this in T cells. Or different types of cells. So here we have the 293 T cells and the K562 cells, and what we've done is taken a yellow fluorescent protein plasmid and then transfected it, created the complex with the peptide and transfected it into the cells, and you can see the signal right there. And if you quantitate the signal out to 14 days, you see that you get a fairly stable transfection. So where is this useful? Well, you've all heard about CAR T cells and the remarkable efficacy that's been seen with the CAR T system. So CD19 CAR Ts have been used in hematologic malignancy treatment pretty effectively, but they do have some limitations. One is you need a retroviral vector to create the receptor, the CD19 CAR, and maybe that has some effects down the road in terms of retreatment or immune response, etc. We don't know, but that's a potential limitation. And so here we're trying to do the same thing, but without a viral vector, so using the peptide. So that's, and we're able to do that. We take human T cells, and take the corresponding plasmid for the CD19 receptor and transfect it into the cells, and you get a very good transfection, and out to here day 10 or day 12 or so, you can maintain that stable transfection. Viability of the cells is very good. The process is relatively non-toxic to the cells. So this is useful because the typical way, again, viral transfection is the common way to do this. And other manipulation methods use electroporation, etc., which can be toxic to the cells as well. What about mRNA? Again, the peptide seems to be pretty effective across different types of payloads. And we, of course, use different peptides for the different payloads. Like, for example, for the plasmid, we would use, because it's the structurally very different, we would use a different type of peptide. But we can find a peptide that works across different types of payloads. So here, this is mRNA, looking at luciferase in different cell lines. And you can see compared also to the standard, one of the standards you can buy off the shelf, which is RNAi Max, which doesn't work that well. And you can see the luciferase signal in these cells, and this is the control untreated. So you can get effective mRNA delivery. We've also done microRNA, in this case MIR34, and which is a tumor suppressor, and you can see that activity corresponding to the dose. And again, compared to RNAi-Max, which is again sort of off-the-shelf, like lipofectamine, we see much greater efficacy in this system. So to summarize quickly, the Aadigen, the delivery platform, has some key features. These are patented, designed peptides that can complex with different cargoes, and you select the type of peptide for the particular cargo that you want to deliver. These seem to be effective in the early testing across different types of nucleotide payloads. And we can also deliver proteins, which we didn't talk about here, but we'll talk about a little bit in the next talk. It's a non-endosomal pathway of entry to the cell, which is important, so there's no endosomal entrapment. And it's an efficient and relatively non-toxic process. So I think I will end there, and that's Pacific Palisades in California.
Thank you. So can we please go into the presentation of Professor De Vita, please?
So do I get some bonus time because I ended early?
Yeah. I think we finally catch up with the time.
Okay, so here, so I'll have a few more slides in this talk. How's that? So here we're going to use the same type of peptides that I just talked about, so I won't spend too much time on the introduction. But here we're looking at gene editing, specifically CRISPR-Cas9. So all of you have heard of this. There's been a lot of noise about this in the last couple of years. And it's a way of editing genes, so either deleting or modifying or changing or correcting genes. And this is a picture of the Cas9 protein, and then here's the DNA and the corresponding target gRNA that's used. So we looked at the ability of the peptide to be able to do this process, gene editing, either via delivering directly the protein, Cas9 protein, which is the nuclease that works, or by delivering the mRNA for Cas9 so that you get it into the cell and the cell produces the Cas9 protein. So we have two ways of doing that. And again, not to give you a lecture about CRISPR-Cas9, but the concept is if you need to delete a gene, you can essentially knock it out by finding the corresponding gRNA to the sequence that you want to cut at. Or if you want to correct a gene, you can insert certain fragments and make a correction. So it's a powerful tool, and it's being used pretty much more or less in every lab in the last 2 or 3 years. So the question is, therapeutically, how do you make this effective? You need to get it into the cell. So you either need to get the whole protein into the cell, or you need to get the mRNA that will make the protein in the cell. So like many other problems, you have the delivery issue. So clearly you have viral vectors which can possibly do that, but we're trying to stay away from viruses and viral vectors for obvious reasons. You have different non-viral vectors, and there's lipid-based systems, polymeric systems, and they have their limitations as well. And then there's the physical methods, which is electroporation, which is essentially opening the pore, exposing the cells to the electric field. And it's not very healthy for the cells, and you can't do it repeatedly. So once again, I won't go through this, but we can use the appropriately selected peptides to do this work. And it can get through the cell membrane by translocation through the cell membrane, not endocytosis. Here's an example of just a size distribution that we see. Again, this is a crude mix. And the idea to show you this, I don't want to show you the fully refined form, final size distribution, but when you it's a self-assembly, and when you put the components together, that is the peptide or the RNA or the protein, Cas9 protein, then you can create a fairly nice distribution. There are some large particles. I don't know if I can click on, is there a way of clicking on that to play the video or no? Anyway, we use, thank you. Again, you can see the Brownian motion. It gives you an indication of size, but these are roughly particles. This is not on a microscope, but this is on a Malvern instrument that's measuring the light scattering. So the resolution is higher than usually an optical microscope. Anyway, we're creating these particles and then using them for different gene editing purposes. So again, conceptually, does this work in vitro? So we take the Cas9 protein in this case, gRNA for green fluorescent protein, because the cell lines that we're going to test them, they're all GFP labeled cells. And then we just look for a knockdown of the signal. And across these different cell lines, you can see that once we treat them, you get a pretty robust knockdown with what we call Aadigen peptides and these complexes, and relative to RNAi max, which doesn't work very well in the system. And if you look at a dose response in one of these particular cell lines, you see that it's effective at a very low dose of even a 1 nanomolar, you're getting almost 70% knockdown. So these work very well in this system. And remember, this is now using the Cas9 protein delivery directly. We can do multiple gene editing at the same time, so simultaneously. So you pick 3 different genes, and these are just model genes that we picked: EMX1, AAVS, HPRT, and these are different cell lines we tested across. And so all of them are essentially complex to the Cas9 protein and put into this nanoparticle and delivered to the cell, and then you measure the indel frequency, which is essentially the frequency of cutting the gene. And you see pretty high levels, between 60 to 85% with this system. Relatively speaking, the RNAi max doesn't work that well in this setting. So back to the adoptive cell transfer. So we can use this in CAR T cells as well. And I think you all know, if you've read about this, that the CD19 CAR Ts work quite well, but they have their limitations. There is often T cell exhaustion after a period of time, and this is often due to the PD-1 checkpoint. So the PD-1 is a receptor on the T cell which the tumors use to essentially shut off the immune activity. So if you knock out the PD-1, that's useful. You can potentially have better activity. Also, the other aspect is, in these processes, they're all autologous T cells. So you take it from the patient, put it back into the patient. So it's a cumbersome process. Creation of a universal CAR T cell would be very useful. And you might have read some things about this, but essentially knocking out the T cell receptor or beta-2 macroglobulin, which is an integral part of the MHC1 complex for recognition, would be useful to prevent graft versus host disease. So that's what we're attempting to do here. So here's looking at the T cells where we're knocking out PD-1. And it works very well. We use one of the standards, which is electroporation, as a control. And you can see that the effect is pretty robust. So you can get about 60% or so knockdown of the PD-1 in the human T cells. We can do the same thing with beta-2 macroglobulin. And again, here we use electroporation as a control, which didn't work that well. But when you use the peptide with the protein and the corresponding gRNA for beta-2M, then you get about a 70% knockdown of that receptor. So, and then here's an example of us. We create the CAR T cell, which I referred to in the previous talk. So use the peptide with a plasmid for encoding for the CD19 receptor. So you've created the CAR T cell. And then you do another step where you knock down the PD-1, for example. And then in this case, you get about a 50%, maybe a little bit greater than 50% knockdown of the PD-1 signal. So we're still optimizing this, but the point being that you could do multiple manipulations and not really hurt the cells. So what happens now? So that was in vitro. Gene editing in vivo is certainly an important consideration, and there's a lot of debate about that. but we wanted to see how these peptides behave in vivo. And again, depending on the peptide, so here we have 3 different peptides, and what we've done is taken mRNA for Cas9 and injected that intravenously into mice. And then we look for Cas9 expression in the different tissues as a way of determining the biodistribution of where the peptide or the particles ended up. And so depending on the peptide, you get a different distribution. So in some cases, you have high in the liver or low in the liver. You have different distribution in the lung. In the case of this peptide, you're getting it into the brain at a fairly high level below the baseline signal, or you're getting into the lymph node. So the point is that depending on the application, you could modify these peptides or select these peptides to go to a certain place. And here's an example of looking at the liver. So this is a model where there's constitutive expression of luciferase in the animal liver. And our goal was to knock that down and see if we could make that work. So we used the appropriate peptide which homes into the liver, and with the corresponding gRNA for luciferase. So we want to kill the luciferase signal. So here's the control, essentially. And then when you treat it, you see a loss of the signal. So when there's several different controls in here, but the bottom line is that when we treat it with the correct peptide and the composition of the particles, you can knock down that signal by about 60% as quantitated by fluorescence. So it works in vivo as well. Here's a sort of a more of a practical model maybe. These are mice, and the idea was looking at cholesterol signal. So you have PCSK9, which I think you're all familiar with. It's a good target to go after. And so we did the mRNA, Cas9 mRNA in this case, not Cas9 protein, with the gRNA targeting PCSK9, with the goal to essentially edit out the PCSK9 to the extent that we could. And so here's the PCSK9 level in these mice over time. And then when they're treated with a single injection we use 2 different peptides just to see how it would work. And you see a pretty good drop in the PCSK9 signal, which is maintained out to the length of the experimental time, which was about 20 days. And so you've essentially killed that signal, or part of that, a majority of that signal. Now the question is, does this translate into cholesterol level? And indeed it does. And you see that correspondingly the cholesterol level has come down by about 45%. In these animals and maintained through the end of the experiment. So again, conceptually, you can do this in vivo. The obvious question is, okay, what are the off-target effects? I mean, we have to determine all of that, but I think we have a good proof of concept that you can use this technology in vivo. So to summarize, again, so these are short, patented amphipathic peptides. We can create these particles, and depending on the peptide, they can be neutral or they can be charged. They're more or less in the 100 nanometer size range, some below, some above, depending on which peptides and cargoes. We can deliver Cas9 gRNA complexes, whether it's the protein itself, the nuclease, or mRNA encoding the nuclease. And we've done that in mammalian cell lines, primary cells, T cells, and in vivo. There's minimal toxicity that we see. I didn't show the data. Actually, I have a slide, but I didn't show. In terms of cytokine release, we don't see very much at all. So it's very manageable, at least in mice. We'll see what happens when we go to the higher animals. So far, so good. And we're looking at different clinical targets to go after. So I'll stop with that. And thank you very much.
Thanks very much. So let's move on to the